Welcome everyone. This is the first research symposium that's being hosted by the Division of Molecular Pathology and Genomics. So thank you everyone for attending. So today we have our keynote speaker, Dr. Muneesh Tewari. Muneesh completed his residency, his MD PhD from University of Michigan in 1997. He went on to do a residency in internal medicine at University of Michigan, and then did his fellowship in Hem-Onc at Dana-Farber. And then he joined Fred Hutchinson Cancer Institute at Washington, Seattle in 2005. In 2008, he was one of the first people to publish on circulating microRNAs and showed that microRNAs released from cancer cells can be identified in the bloodstream. And that has subsequently led to this whole body of research on using microRNAs for early detection of cancer and various other applications. Muneesh moved to the University of Michigan, moved back to the University of Michigan in 2014, where he's currently an endowed professor in the Department of Internal Medicine and Biomedical Engineering. His research interest is not focused on any specific type of cancer, but rather his interest is really in discovering new biology, developing new technologies, all with the goal of early detection of cancer. His more recent interests include investigations into the role of human consciousness in human health and disease as well. So welcome, Dr. Muneesh Tewari, and please give him a hand.
Thanks very much, Bharat, for inviting me. And I should say it's really a special honor because, oftentimes for these kind of symposiums, people invite people they know. I actually didn't know Bharat at all. This is like my first time at the University of Minnesota. So I assume that means that this invitation was actually from, looking at the papers and some interest in the work that's actually done. So, so I really appreciate this opportunity to speak to you all. And as Bharat said, my group doesn't really necessarily focus on one cancer type or one disease. The title more or less says it all. It's really about new approaches. How do we think about disease detection? How do we think about disease monitoring? And again, we do focus though in the oncology space, and that will be reflected in the talk. So rather than going really deep in terms of particular data on one type of cancer or one type of biomarker, this is going to be more like a little spaceship ride, a little bit into the past. It's sort of my own personal experience in this field of biomarkers and liquid biopsy in particular, since Bharat mentioned that there was quite a bit of interest there. And then a whole bunch of stuff about the future, and some of it's kind of sci-fi about where things might actually be going. So I'll just flip ahead here. So liquid biopsy, I'm sure, I imagine, most people in the room are familiar at least with the concept, but the basic idea is pretty general. It's the idea that tumor cells are shedding materials into the bloodstream, oftentimes as they die, sometimes potentially through a secretory mechanism. And of course, normal cells are also doing that. And the basic idea is there's all kinds of different analytes that might be detectable or measurable in the bloodstream. And so that, instead of doing a biopsy to get information on the cancer, or really, people sort of, even the slide says versus, but it's not really versus. It's more like in addition to doing a biopsy or to complement a biopsy, or sometimes to actually have it as an alternative. You would look in the bloodstream, and obviously, the sampling is easier, and you can do this over time and all of that, and that you would actually get data. And as you may have heard, there's all kinds of different liquid. Again, this term liquid biopsy is probably over the last 10 or so years. At some level, really, this is blood-based biomarkers, and people have been doing this for decades. at some level of PSA measurement is a type of liquid biopsy. But what's been really exciting, of course, is that the degree of information that you can get has really expanded, with sequencing tumor DNA shed by cancer cells, etc. And there's lots of potential applications in this field, so everything from early detection, can you find the cancer, can you determine where it is, has it spread outside of the localized site, what are the somatic mutations in the tumor? This is sort of one question I think that's probably advanced the most clinically. What treatments might work, are the treatments working, and is there any residual disease after an initial treatment? And so this is all very exciting, and the idea here is that there's a potential of these kind of approaches to give quite a bit of extensive information and relatively non-invasively. So I am going to start really by telling you, look, we're going to jump into a little bit of history, and again, it's going to be personal history because that's what I know best, since I lived through it. So one of these, of course, you can look at multiple different possible analytes. Only some of these are listed here. These are some of the ones probably I would say over the last 10 years have been extensively developed. The idea of DNA from dying cells, of course, the idea of cells themselves shedding from the tumor and going through the circulation. But there's also, as Bharat mentioned, a whole field of potentially of RNA. And I'm going to take maybe the first 10 or 15 minutes to talk a little about the RNA, not so much even specifically about this. The purpose of this isn't necessarily you'll get a little informed about the RNA as a biomarker, but more really to give you a sense of kind of like what I've learned, picked up, trying to, basically being a researcher in this field. And I didn't start out really in biomarkers or diagnostics or any of this. My postdoc was actually in systems biology, in nematodes and C. elegans, and in yeast. And so when I became a faculty member in 2005 at Fred Hutch, I really got, I had switched to working on microRNAs in cancer. So I had sort of migrated a little bit, but I got inspired by Lee Hartwell, who was our director at that time. And even though Lee got the Nobel Prize for really deciphering the cell cycle in yeast, by the time he was a director here, he was very interested in early detection. And at the time, there was a big push on proteomic approaches. We had rooms and rooms of mass spec equipment and people that Lee had recruited. And he came to one of our faculty meetings and said, hey, why don't you all think about whatever you're doing, and maybe in some way it can help early detection. You all have assays that you're running in your lab and so forth. And I think a lot of the faculty in the room, probably the more senior faculty, just said, oh, okay, kind of rolled their eyes. Okay, there goes Lee again, maybe. I don't know, I'm just imagining, or it just sort of went in, 1 year and out the other. I don't know. But all I can say is maybe because I was young and naive, that sort of registered in my brain. And we were working on microRNAs at the time, and we decided to just ask this question of could microRNAs be there in the bloodstream? And at the time, the real limiting factor with proteins was sensitivity. There was no PCR for proteins, so you needed a lot of protein. And so how are you going to do early detection of small tumors? Whereas microRNAs being nucleic acids, of course, could be amplified by PCR. So I naively thought that these microRNAs might be so small that they might not get degraded by all the ribonuclease that's in the bloodstream. And this was one example of where being naive actually turned out to be a good thing, not always the case. And long story short, I'm just going to flip through this stuff because the details don't really matter. It's more, I think some of the takeaways. As Bharat had mentioned, we found that we could actually find these microRNAs in the blood, in the plasma. If you measure 3 microRNAs on plasma sitting at the bench for 24 hours, they were incredibly stable. We ended up working out why they were so stable. It was protein complexes and sometimes extracellular vesicles like exosomes. We did a bunch of profiling, sort of more genome scale microRNA profiling, and found particular microRNAs, in this case advanced prostate cancer patients, which were many fold, 10, maybe 40-fold higher in their serum compared to healthy individuals. And we worked out some of the mechanisms. They regulated, in this particular class group, epithelial differentiation. We found other microRNAs that were regulated by hypoxia, and that made us think, well, maybe these are this could actually relate to tumor resistance, because hypoxic tumors tend to be resistant to therapy. This is just summarizing years of work, and we found that's true, that there are this particular microRNA was elevated by over like a log-fold higher in patients who weren't responding to therapy compared to ones who were. And so I'm really summarizing like many, many years of trying to take this idea of, hey, here's a new class of biomarkers, and hey, we might solve early detection with this. And that was very inspiring in the beginning. And then, we went through this. We went through a whole other series. Every one of these fields I've noticed, whether it's proteomics or metabolomics or whatever, they all go through this series where pre-analytic, which I think people in this room totally get, as well as analytic challenges have to get resolved. So we figured a bunch of that out. And I have to say, laboratory medicine, Colin Pritchard was in my lab for a bit. He's a laboratory medicine now professor over at University of Washington. And he really helped us actually work all this out. And that all being the case, this is just another example of analytic challenges. We found out that digital PCR, is probably a better platform than real-time PCR and so forth. But what I found, though, is that after, about a decade, really, of working on this, and after having about 3,000 papers, now I think it's probably 4,000 published, in this area, it was all great, but there really weren't very many, actually there were none, no microRNA biomarkers, liquid biopsy based on RNA, even close to the clinic. I mean, we had a couple that had gone into early, like SWOG, multi-center trials, but nothing really, really close. And I must say that after a few years, the last, maybe the first 3 or 4 years, this was all very exciting. In the middle, there was a little bit of, what's going on? And then after a couple more years, it was kind of like, well, I guess the funding so far is coming through and we're publishing papers, but something's not really adding up here, and is this really going anywhere, and what's the point, if it's not? And so that really prompted me to do some, honestly, self-reflection, some conversation, some reading, some thinking, and I'll just summarize for you in the next few slides, like some of the things that I learned from this process, because I think they're relevant to liquid biopsy, to disease monitoring, to all kinds of methods just in general. So why was this not happening? Well, one of the reasons, I think, is that ultimately, if you do clinical translation, and again, to people who do this, this is probably very obvious, it's every day, but again, I came into this not having done any type of translational or clinical research, and then sort of grew up, as a faculty member trying to learn this, and the bottom line is, it's a tough problem, right? It's like you have to line up all these apples, and you have to shoot one arrow all the way through them that links all of them together. So you got to have the right biology. And I think we were on the right track with the microRNAs. I mean, they were amplifiable. They were released from tumors. They were regulated by tumor-related processes like hypoxia. Then by samples, what I mean is, not just the clinical samples themselves, but also the right kind of sampling and processing and the right kind of technology. So I feel like we made a lot of progress on a lot of this. But then you got to thread that needle or this arrow also through the unmet clinical need. And also then there's a societal component. So what I mean by this is obviously, people sometimes say it doesn't make a difference. I always like to insert positive because sometimes it can make a difference, but it could be negative. So we got to make sure that's the case, right? And for every 10 biomarkers, we would discover maybe only one really actually threads that needle to the need that's going to be actionable. And I think the same principle is coming up with circulating tumor DNA, where there are a lot of companies who are able to sequence circulating tumor DNA and tell mutations in blood. And that's great, but one of the questions people are asking is, well, where does this go next? How do we expand this? Well, it's related to, well, is there really a need that the biomarker is going to plug? And so, some of it was this. And then, of course, there's this whole other piece, which is you can even thread the needle through the unmet clinical need, but you still got to address who's going to pay for it, both for the initial development, the tens of millions of dollars to actually do those studies that demonstrate that it fits a clinical need, and then, of course, the risks, the FDA, or the CLIA lab, or whatever path you take, and patient adoption. Anyways, I mean, I don't want to, due to the limited time, I won't go through all the details, but I've certainly got stories about how good biomarkers have failed because one or more of these have not been addressed. So this is one of the things I guess I took away, from that decade. I would say is this image right here, and the other thing I took away. Well, some of this is just patience. In that, I don't know if you guys have seen this this thing called the Gartner Innovation Cycle. It's also called the hype cycle, and if you read it, it's like you get some new technology. It could be technology, but it could be anything new in your field, right? Then you get this peak of inflated expectations. I definitely had those. And then you have this trough of disillusionment, and I can tell you I went through that too. And then you get this slope of enlightenment and eventually a plateau of productivity. But the funny thing is, by the time you get here and the stuff is actually working and making a difference, nobody even notices. It's like nobody cares. Oh yeah, of course that's happening. I'll tell you, gene therapy is a good example of this. When I was a MSTP student, like I started in 1990, it was the rage, and it was everybody thought gene therapy is the thing to do and it was going to cure everything. And then of course it crashed, as everybody knows, probably early 2000 or whatever. And now they're doing gene therapy for some pediatric diseases, but never hear about it, right? But that's okay, because that means that's a sign of success. So in any case, so some of this was maybe this, we were just going through this cycle and now we were sort of coming up, hopefully, out of it. But I have to say, it didn't seem like this might explain all of it, because if you look through sort of the late 90s to current time, there have been a lot of new technologies, the whole genomics, proteomics, metabolomics, just as examples. These have all really flourished in this time period. And there's also new types of analytes. Cell-free DNA, RNA, these things have been, maybe people have poked at these several decades ago, but really it's been the last 10 or 15 years that this has really come into the mainstream. But yet again, it's not just microRNAs that we're suffering, just in general, really on the whole, trying to get biomarkers that work in individuals. So, cholesterol, lipid profiles, these are biomarkers, they work in populations, right? But they don't really work in stratifying very individual risk very, very accurately. And again, I wondered, what is the reason? And so some of it could be this, it's just hard, you got to line everything up. And some of it could just be, well, it takes time and you got to go through ups and downs. But again, people have been doing this for, a while now. And there's a lot of investment, both on the public and the private side, that's gone in. And so I kept thinking about, is there something more fundamental? And I'll share with you just, my own opinion or ideas about what this more fundamental thing might be. And it's actually pretty simple, conceptually, but practically, it's maybe another story. And when I really looked at this, some of the reasons for failure are a lot of the biomarker studies were not well designed. Again, not good attention to pre-analytic or analytic variables. Sometimes we didn't know enough about biology. But this piece I didn't really see, I haven't seen people talk that much about. And this piece is just about our fundamental assumptions of how biomarkers work, of how we predict things. And I would actually argue that in most cases, the biomarker problem or the liquid biopsy problem is really a prediction problem. Even when you're trying to do early detection, at some level you're trying to take a blood sample and then predict what the imaging test that might follow up looks like, right? Or you're trying to predict from the blood what the tumor biopsy might look like. And it turns out that a lot of times, our traditional approaches are not taking into account the nature of biomedical systems, or really biological systems, that they're complex dynamic systems. And normally I have a few slides that actually delves into this, but again, because of time, I took them out. But the bottom line on this is that there are simple systems. That's a few interactions, and they're all sort of linear and predictable. There's complicated systems, which can have a whole bunch of things, and then there's complex systems. And biology is complex, and what it means is there's lots of interactions, right, and they're all nonlinear. And when you get to complex systems, making predictions is actually really challenging, not using our traditional approaches. And so, again, this nonlinear dynamics, if this is really underlying what's happening, and I think there's good reasons to think that it is, what this means is that if we look at any sort of disease generically, if this is the activity of the disease and this is time, well, if there was sort of a linear relationship, like the disease builds up over time, that's fine. You'd get something like this. But if it's really a complex system, they're always nonlinear relationships. And especially in biology, as I'm sure people are aware, the sigmoidal curve, for example, is very, very common, right? Hemoglobin dissociation, DNA melting, E. coli population expansion, influenza spreading in populations. All of that is this kind of curve. So what that means, again, thinking about this very conceptually, is that early on, if you're trying to do a detection of some disease or some toxicity of a therapy or anything, any significant event, the signal is pretty low because the underlying biology is not that active, and then relative to the noise. So that's a hard problem. But then if you wait long enough, the signal gets super strong, and if you just take one blood sample, let's say, and you compare it to a controls, it's pretty easy to see it, but usually by that time it's maybe too late or beyond when a clinical intervention would make a difference. And this is true really in all kinds of complex systems. Again, whether you look at ecological systems, sociological systems, a great example is the weather, right? So making a prediction of what the weather is going to be a year from now isn't very accurate, but if you collect data every day or every hour, you can make pretty good predictions of what's going to happen, in the next day or in the next few hours. Although I don't know, I haven't seen the Minnesota weather people on TV, so maybe it's not true here. I don't know, but that's the basic idea. And this thinking has really, I guess, sort of entrenched itself in my head. And so the basic idea is that right now, generally speaking, this is a generalization. There are exceptions, but the basic paradigm is that you have a person and you have a reference range, right, from a group of other individuals, and that defines what's normal. And you take a blood sample and you compare it to whatever the control group historically is, and then you define, what's normal and what's abnormal. Now the challenge is, of course, when it works, it's great, but there's a lot of day-to-day variation. There's a lot of person-to-person variation. What medications are they on? All kinds of things. And it's just impossible to study all of that for any biomarker. I mean, it's just, even for PSA, I actually did a deep dive into how much do we know about, how does variation, genetic, epigenetic, environmental, lifestyle affect PSA, and it's, like, we know very little. I mean, even for something that's used, whatever, millions of tests, probably many more than that over time. So the alternative, of course, is really simple, but maybe the challenge comes in how do you implement and how do you really think about this moving forward, is just to take a measurement within a person, high time resolution, and then now you've actually got more like a movie instead of a snapshot. And I know that this is being talked about, at least in I think lab medicine circles, because Colin told me that there are people looking at these as patient-specific reference ranges as one example. So this is the basic principle, and I would say again, the core drive from a biomarker and monitoring liquid biopsy perspective in my lab now for the last few years has been, how do we actually do this? How do we test this idea that actually moving or at least having this additional paradigm to complement what's already happening would actually be useful? And the biggest challenge I saw initially is just, well, how do you get all this data, right? Because, I mean, it's just expensive. The patient has to go to the get a phlebotomy, and has to go to the clinical lab, and it's like, even if it's $20, $30 per data point, if you want to get like 20 data points, this all adds up. Even just to do the research, it's really incredibly expensive. And this actually, is one of the things that helped prompt my move actually to the University of Michigan, because I started to talk to engineers, actually even in Seattle and other places, about how could this be possible. And I have to say that this slide is actually from like 5 years ago, and maybe even 6 or 7 years ago. And I started, we started to think like, oh well, maybe someday you'd have a glucometer, that could measure not just glucose but all kinds of other stuff, and it would be synced to your smartphone. And you can see this is a older version of the iPhone, much older, And the funny thing is, this is still sci-fi, but the striking thing to me has been, since I came to Michigan, like very, it's just people have come out of the woodworks in a way, and the engineering and other physical scientists, And so we have a project. It's too early right now to actually tell whether it's their success or not, but there's a collaborator, Jin Sun Kim. He's a materials scientist and engineer, and the idea is, can we repurpose these glucose test strips to actually measure nucleic acids instead of glucose? Because it turns out that they have glucose oxidase in here. It liberates, basically FAD is a cofactor for it. And so anyway, so he's also an organic chemist, so he's come up with some compounds that we hope are going to be able to, when they react with when the right nucleic acids basically hybridize to each other, the two compounds are conjugated to basically two probes, that it would liberate FAD, and then that will actually ultimately be able to be translated. So this is a way to sort of leverage all of this technology that's already there, pretty cheap. These are like $20 or whatever, right now at Walgreens. So this is just kind of an example of a thread. We're not the only people doing this. There's a lot of work going on, honestly, nationally, internationally, of trying to develop really cheap and inexpensive ways of doing these measurements. And when I think about it, it's like, well, the technology is evolving. There used to be the iPod, there's the iPhone, there's the iPad, and what we hope is that with these kind of things, our next evolution will be the IP. And I thought this was just kind of a joke and kind of funny until, again, we ended up, once you start talking to engineers, humor gets weird, everything happens a little bit differently. So we have, we actually started to work on a little bit of a project to see, can you actually put something, in the toilet that will just monitor urine on a daily basis. And what I learned really from that is the technology is, believe it or not, the easy part right now. The technology is doable. It's like if you had a big enough why, that would attract the money, and you could do that. But the missing part right now is still the biology and the demonstration that, okay, there are analytes that it would make sense to measure over time, and that would really make a difference, in terms of really a clinical application, the actionability. And so that's where we've been focused. So these are nice threads that we've been pursuing with collaborators. But a lot of our effort now in the last, again, maybe 3 years or so, 3 or 4 years, has been focused on that piece. So what are kind of, maybe not even necessarily technologies, but what are the approaches that we should be thinking about that could get us this kind of data in the near term that's time resolved, and what are the contexts so that we could actually test this idea that, wow, this would really make a big difference in terms of clinical practice. And of course, getting like an FDA-approved test, that's way into the future. That's not really where a lot of our focus is right now. It's really, again, the new approaches, and does this idea really make sense? Does it actually have any legs to stand on? But you get the idea here, because I think the real benefit of this would be passive, unobtrusive, monitoring, and that kind of data. And so now I'll actually bring you back to cell-free tumor DNA. And this, so I'm really going to share a couple of stories really with you. One is going to be on cell-free tumor DNA. The other one's going to be very early, very, very early, but it incorporates wearable sensors, because Bharat had actually asked, and it seems like there's some interest in that. And so that second story is really about how we're thinking about disease monitoring kind of moving forward. And this story is really about how we are trying to think about how circulating tumor DNA, this approach could be improved and to overcome certain barriers, and also could be used to enable this, again, this time-resolved monitoring. Now, the basic idea of this, again, I imagine a number of people may or may not be familiar with this, but this idea of in the blood plasma, looking at fragments of tumor DNA released by the tumor, has already had some clinical success. And so there's an FDA-approved test in lung cancer patients to look at for particular EGFR mutations that have some actionable information. There's a number of companies like Foundation Medicine, Guardant, and others that are doing laboratory-derived tests, essentially, where you can send a blood sample. They will sequence, anywhere from dozens to a few hundred genes from the cell-free tumor DNA and identify what mutations are found. In some cases, it's being used as a surrogate for for biopsy, because for example, in lung cancer patients, sometimes the biopsy will be inadequate for doing this kind of sequencing. So this stuff is already happening, right? But most of the application right now has been as really determining what's the somatic mutational spectrum of tumor tissue in a patient. But moving forward beyond that, I mean, there's lots of other applications that liquid biopsy could really fulfill in theory. Like applications like after giving a definitive treatment and there's no cancer visible on any imaging, well, are there any cancer cells left there? And so, minimal residual disease, another phrase for that, right? Or after a patient, again, has no evidence of disease on any imaging, over years, they're at risk for a recurrence. So could we detect a recurrence earlier? Or once a therapy is started, and some of the newest, many of the therapies, as you probably know, in oncology, can be pretty toxic, and even some of the newer therapies like immunotherapies can be pretty toxic too. So once a therapy is started, can you know sooner whether it's working or not? Now, these are actually challenging problems using the current approach, and it's for a couple of reasons. So one is, in all of these situations where I described, the disease burden is actually pretty low. So the current successes with blood ctDNA are mostly in stage 4 cancers with a higher disease burden, a little bit in stage 3, but once you get to stage 1 and 2, the sensitivity of just detecting any tumor DNA in the periphery is actually pretty low. And so that's one challenge. And then another challenge, of course, is if you want to really look at tumor dynamics with high time resolution, there's some limitations on how many times can you actually draw the blood, both in terms of what's safe, but also in terms of what's practical if you were to deploy this, on a sort of population level. And again, I think the disease, when the disease burden is low, the challenge there is that the tumor might shed some cell-free tumor DNA, but it gets diluted in 5 liters, of blood volume. And when we actually sample, even if you took like 40 ml of blood, 4 10 ml tubes, it's less than 0.5% of the total blood volume. So if there isn't enough tumor DNA there, what can you do? There's not a great solution immediately. So an approach that we've been working on is this idea of what if we tried to measure this tumor DNA in the urine instead of the blood. And the idea here is that it's actually transrenal tumor DNA. So the idea is that the blood, of course, 5 liters of blood volume circulating through the kidneys, goes through the glomeruli, back into circulation. This is happening, and that's, in a day, I forget what the numbers are, but many, many times you're circulating your whole blood volume, filtering through the kidneys. And when that happens, the idea is that some of that tumor DNA actually gets through the glomerular barrier. And there was data hinting at this, at least. I mean, in the academic literature, there are a few papers from about 15 years ago indicating that this happens in cancer patients. There's stronger data in pregnant women, showing that fetal DNA, gets in there, gets into the urine. And there was even a company, Trovagene, that was marketing a test specifically for a particular cancer mutation, EGFR, using this approach. So about 3 or 4 years ago, what I was struck by is that there just isn't a lot of academic data, or in kind of the public domain of, is this really real? If it's true, how much DNA is there? Like, how common is it, across different cancer types and all of that. So we really invested a fair amount of effort, and we actually have not published this yet, so it's all unpublished stuff I'm going to show you, but hopefully we'll get it published soon. But we really tried to really dig into this and see, is this a real phenomenon? Does it have potentially value in trying to overcome some of the current limitations of cell-free tumor DNA in blood to really get to the next level of applications? And so, so I think I already talked about this. And again, basic idea is tumor DNA gets into the blood, and some of it gets to the glomerulus and into the urine, and then potentially collects there. And that would be in theory. And of course, some of the advantages, this is now truly absolutely non-invasive. I think the big advantage also is you can collect large volumes. And we've been now doing these studies where we send patients home with like 400, 500 ml bottles and with a UPS, sticker, and basically they collect them and they put them on their porch, and the UPS comes and takes them and ships them. And I was worried that I don't know how practical this would be, but it seems to be happening, you know. And so far, we haven't had too much trouble with this. We also had another study trying to do the same thing with stool, and that was a lot harder. So I guess, you know, so far so good with this. So I'm going to just give you a few slides to just show you what we've seen with this. So the first thing we wanted to do is, you know, even though there were these papers, again, they hadn't really gone in depth with the methodology, like they were doing a PCR of a KRAS mutation and running on a gel and saying, oh, we got a band in the cancer patient and not in the non-cancer patient. So we wanted to, you know, I didn't know what to make of that. I mean, this was fine for 15 years ago, but in the current day and age, I felt like we had great access to methods where we could really know more definitively, is this a real phenomenon and how significant is it? So the first thing we did was to say, let's go to some acute myeloid leukemia patients, because leukemia patients have circulating tumor cells, you know, in addition to circulating tumor DNA. So there's quite a amount of DNA in the bloodstream. Because if we didn't see it in the urine there, then, you know, it's probably not going to happen, you know. And so the first thing we did was we decided to do whole genome sequencing of the DNA from the urine. So we just shotgun sequenced DNA, you know, urine DNA. And, I mean, we had to use a bunch of tricks because the DNA is fragmented and all that. I won't go into those details. But then we aligned those sequences to the genome. And, of course, you know, if everything is diploid and there are no chromosomal aberrations, you should just see, if you plot these alignments, you know, chromosome 1 to chromosome 22, which I'm about to show you, they're all pretty much at the same level. But actually what we saw, first I'll show you in the plasma. So this is a patient with this is what the cytogenetic karyotype was, you know, clinically, that was read out. And then this is the plasma of the patient. What you can see is, you know, there's some things like loss of chromosome 5, and you can see that, you know, gain of probably chromosome 8, you can see that. And what we were really kind of quite excited to see is that in the urine, you could also see this. Now, you might say, well, it looks like a little bit cleaner in the plasma, but that's also because the whole bioinformatic pipeline was actually optimized for plasma, where the DNA is longer and the DNA is shorter here. But the basic idea is, you know, we could read much of the karyotype of this patient's leukemia, you know, from just shotgun sequencing the urine. Now, I'm not advocating that as a clinical test or anything like that, but as our proof of concept, you know, really convinced me that, oh, okay, this sounds a little bit far-fetched that the urine DNA, the blood DNA would get through the glomerular barrier into the urine, because again, you know, the glomerular barrier is the glomerular barrier, and the DNA in the bloodstream is believed to be mostly in histones and all of that. But the bottom line is, we actually saw that this looks pretty real. And I will tell you that, you know, we did this in then a whole slew of different 13 patients which had known abnormal cytogenetics, and they had at least 10% peripheral blasts. So we haven't, you know, this is all pretty early research, so we haven't now done the next study of going, you know, to lower disease burden. But basically in every one of them, you know, we were able to detect these copy number alteration. And then we did a little survey of solid tumor patients, and different kinds, lung, rectal, colon, breast, sarcoma. And we wanted to see, you know, for patients who we knew had circulating tumor DNA in the blood, you know, could we see alterations in the urine? And I'll just give you one example. And the bottom line is, you know, we could see that, you know, in throughout the patients. But here's one example of this rectal cancer patient, because we had sort of samples, a few samples over time. And you can see here's the plasma. You see all these chromosomal abnormalities. Here's the urine. And these are pre-treatment samples. And what was, you know, kind of cool to see is that this is day -1 pre-treatment, the urine. And then once you get to day 14 and day 22, and this was a patient who actually responded to therapy, you know, the profile normalizes. So again, you know, this kind of thing has been done before in blood, but first time we're really seeing it in urine and suggesting that, you know, maybe this is really, again, for real. So that was been the first thing, you know, for me. The other interesting thing that I think is harder to do with blood, and actually, you know, this has been seen actually using the Trovogene assay before as well, is that we actually saw a spike, you know, at about 7, 8, 9 days after initiating treatment. And I think, I mean, right now I presume that that's actually the cells dying, releasing their DNA, and then we can actually, you know, look at it in urine. Again, the beauty of this is we can actually get urine every day from patients sitting at home, which is a lot more practical than if we were trying to do this with blood. And then the most important and maybe exciting part of this, which is still very preliminary because it's the number of patients isn't like 100, it's more like 10, right, or less, is that we came up with quantitative assays. And this isn't not trivial, but so I won't go into the details, but we came up with quantitative assays where we could actually quantify a number of molecules in the urine and compare them to the blood. It's not trivial because the fragment sizes of this urine DNA are very small, so standard ddPCR assays don't work. But when we did do this, what I was really struck to find is, and this is just the volume of urine we got, like from that particular patient, because we weren't trying to get large volume, and this is 10 ml, and this is again extrapolating the numbers. And for many of these patients, there was, there would be more number of copies in the urine than in the blood. And in some patients, okay, maybe there were a little less, but you can imagine that if you got 2 liters of urine, which again, from our experience has not been a problem, that you would actually get more copies. And it kind of makes sense with the idea that, again, this is collecting in the urine because you're filtering. Of course, there's also breakdown happening, so this is the end result of the whole process. But I think, to me, this is potentially the most important part of this, is the possibility of increasing sensitivity. And there's 2 ways we can increase sensitivity. One way is just by actually getting more molecules. But the other part that people don't talk about, I feel, is that when we sample a patient's blood, we don't know whether cell-free DNA is released episodically or whether it's just a steady state. There's actually good reasons to think that this is going to be episodic, and if you don't catch it at the right time, you might not get you might not actually be able to detect it. And again, with urine, I think if you can take multiple samples, or if you were to take samples over a week and multiple days and average them, or actually sort of compile them, we might have a greater opportunity to do that. So anyways, in our lab, our goal is to try to really develop this as an approach. Again, this is very early stage, so we're developing methodology and other aspects of this. Again, I won't go into it, but we have other collaborators where we're trying to use some single-cell imaging of DNA molecules to really make this very cheap and robust moving forward. So, okay, so this is just a summary, I think, of what we talked about. So the bottom line is, I think it's a potentially worthwhile direction to explore. So the last part of this, I'd really like to talk about how again can we enable this, right? And another phrase for, I think, what I've been talking about that has come up actually in the literature as well as just in common use now, is this idea of disease interception. And so instead of like early sometimes people call it early detection, but it's sort of saying all the same thing. But the basic idea is, instead of reacting to disease, which we do a lot of times because we wait for symptoms or we wait for, some significant abnormality, can we actually intercept disease? And again, I think we're all talking about the same thing, but the basic idea is on this curve, can we identify a moment, right, in time, it might be over here, that's early enough that we can then intervene. And the urine DNA approach that I just mentioned is one approach to start doing that for cancer patients. But, I've been thinking a lot about how much work it takes and how much time it takes to develop such an approach and then get the technology and get validated and all the analytical validation, pre-analytical, all that stuff. Is there anything we can do right now that would be actually faster to try to get this high time resolution data? And that's what really led to this interest in wearable devices. And so, can we integrate non-molecular biomarkers, so the data that comes from a wearable device, considering that as a biomarker, and can we integrate it? And this was actually several years ago now. I think Time magazine had this thing in the future. You just have like subcutaneous, sensors, and your phone will be in your arm or whatever. And I don't know if that will happen, but clearly there's been a huge burst of activity in consumer wearable devices, right? Apple Watches, and again, this is a really old version, and all that. But it's very exciting, I think, because we have now opportunity to get this continuous time resolved data. And for discovery research, which is really where we're at right now, these patterns of time are really not well studied. outside of the ICU, there's continuous data, and then, in exercise physiology, there's this kind of data. But aside from that, like I've not been able to find in the literature really much data on when patients go home, what's happening really moment to moment or minute to minute, with their heart rate, with other parameters. And the other thing I think that would be really exciting was if we could link some of the biology of what's happening with patients and their disease processes to some of these biometrics. And I'm going to show you an example of a study that's just underway. I actually, at the last minute, yesterday, I got one piece of data, so I'm going to actually show that with the caveat that it's raw, unfiltered, unprocessed. But just to show you really, just share, I guess, how we're thinking about it and an example of a study that's underway. And this study in particular, we decided to study patients who are getting CAR-T immunotherapy for cancer. And so CAR-T, some of you may have heard, it's chimeric antigen receptor therapy. Basically, you take T cells out of a patient, engineer them to express a receptor for an antigen that's present on the tumor, and infuse them back in. And of course, like any new therapy, there have been there's side effects and there's toxicities, but also there have been some stunning successes. And so, a very new type of approach. But one of the big challenges with this is the toxicities. So, and one of the big toxicities is really this cytokine release syndrome, where the immune system gets activated and just dumps a ton of cytokines. Patients get hypotensive, go to the ICU. It can be life-threatening, to say the least. And we think that this could be a great opportunity to actually try to apply some of these approaches, and hopefully eventually make an impact in how this whole disease is both monitored, predicted, detected, and then acted on. And so the basic idea here is that right now it's very hard to predict who's going to get this, and it's also very hard to predict when they're going to get it. And so an accurate predictor, if we could actually have a risk score over time that could actually predict, hey, what's the risk of this patient getting this cytokine release syndrome, let's say in the next 24 hours, then that would actually be actionable by intervening with a therapy. So there's actually a therapy for the CRS, but usually, it's applied relatively, it's all relative, but relatively late in the disease course. And because CRS also evolves very rapidly. And right now, all of these patients are, at least at the University of Michigan, are all admitted, and they're in the hospital for 2 to 4 weeks, sometimes longer, being monitored because a lot of times they're just sitting around being monitored, but you can't really send them home because they might develop CRS and die, right? And so that's also very expensive, to be very, very frank here. I mean, it's hundreds of thousands of dollars is what's billed for one hospitalization for this. So our overall approach has been actually, there's sort of two elements to it. So one has been, can we use wearable sensors here now to try to get at this? But the other has been, can we also integrate the wearable sensors with the biological readouts potentially of this? And I won't go into all the details here, but basically, ultimately we're trying to actually mine the health record data. We're getting stool samples over time for microbiome analysis. But the really important, I think, relevant sort of hypothesis-directed things are, we know that cytokines are involved, and we know that these cytokines, some of these actually act also like TNF-alpha, IL-1, on the hypothalamus in the brain. So we have a hypothesis that body temperature regulation will be altered. And our hypothesis has always been, this may not be a fever, it might be a shift in the normal pattern of temperature. We're also looking at heart rate variability activity, again, with wearable sensors in these patients. And then for the cytokines, we're collaborating with collaborators actually in the engineering school with point-of-care devices so that we could actually get cytokine measurements, day to day. I mean, within, the goal like being, within half an hour or less. And so this is a big project where we're trying to do all of this on these patients who are getting CAR-T therapy before they start therapy and until and through after they develop CRS and get treated. And so, not surprisingly, it requires lots of collaborators. So Katsuo is our engineering collaborator, Sung is our clinical collaborator, and the people in my lab and others. And what I'm going to show you, because I don't have, this has just gotten underway, but I have one slide of, again, unfiltered raw data, hasn't been processed or anything like that, on temperature. And this is in a patient who developed CRS, and we got temperature readings every 2 minutes, 24/7, in this patient. And it's just an anecdote, but it's maybe a taste of maybe what's to come. And this is what we saw. So this is over a 3.5-day period. This is actually when the patient clinically over here a decision was made to actually start IL-6 treatment, anti-IL-6 receptor actually. And then the patient went to the ICU here, actually crashed and went to the ICU. And this is the temperature reading. And what's interesting is in the medical chart, at least my research coordinator told me, is they documented the patient started having some symptoms, some rigors, but there was no fever. Okay, but fever is defined in our hospital system as 100.5. You can see the temperature never hit 100.5, right? But what's really exciting to me, and you can ignore these spikes because this is unfiltered data. So when you get really practical about this, and these patients have this wearable sensor under their armpit, if they move or they roll or whatever, you get like this stuff. This is noise. So we would get rid of that. But what you can see is really ultimately there was this rise in temperature. And this temperature rise happened, potentially, certainly before a decision to make a diagnosis of CRS. And of course, we've got to dig into this further. And so this is a very simple interpretation, right? But what we want to do is once we get a lot of data like this is to actually apply machine learning approaches because there may be signals buried, in even the subtle variations of this that could be predictive. So that's, again, just a taste of what wearable sensors might do in the future. And I just want to leave you with the big takeaways. I think this is a big takeaway of how hard translation is and what else you have to line up. Just to remember this hype cycle in whatever you're working on and wherever you're at. Sometimes I find it's good to go back to this and kind of realize where you are on the hype cycle. It can be kind of have a calming influence, The idea that maybe in the future, we might be looking at integrative approaches and temperature and really wearable measurements. And again, this is the research where we integrate all this. I don't see this as being the clinical practice. Clinical practice to integrate all this and then reduce this to the simplest number of factors, right, that are actually making a difference. And ultimately, this idea of, maybe this is a direction we're going. And the last slide before acknowledgments I'd just like to leave you with is a slide with a quote from Albert Einstein, who's one of my personal scientific heroes of why, I think we're pursuing this. And, it's sort of self-explanatory there. Yeah. So I'd really like to acknowledge actually all the patients who give us so many blood samples and wear these devices. I tried to acknowledge actually most of the team members and collaborators in the slides and our different funders. So thanks so much for your attention. Hopefully we have a few minutes for questions.
Questions?
There's one right here. Yeah.
That was a great talk. Thank you very much. I was intrigued by your idea of looking for important DNA in the urine. And of course, this is a tremendous idea. 20% of the total blood volume goes to the kidneys on a daily basis. And there is a filtration barrier, of course, but the limit to filtration is about 60 kilodaltons in terms of size. But there's also a charge limitation to filtration. And so my guess is that a lot of DNA is actually negatively charged, which would inhibit filtration.
Yeah.
But if you could find some way to change the charge of the DNA you're interested in, it would increase the amount of filtration. And of course, 99% of the urine that's filtered is reabsorbed. So if you have something that's filtered and not reabsorbed, you have a huge concentration. So that's a tremendous idea. You want to expand a little bit more on that?
Yeah, I mean, I think, we're just trying to take it step by step, but exactly what you're saying, I think there are potential ways to increase the yield. And so I can show you, I had a couple of extra slides. So if we actually sequence the DNA and actually looked at the tumor-derived DNA, the bottom line is it's about 25, nucleotides or base pairs, which we don't know if it's single or double-stranded. That's the median size. So I think that the larger DNA is actually restricted from filtration. But absolutely, there's reabsorption. And so, we've thought about some maybe harmless, either an over-the-counter types of agents that might be given at some point to actually either loosen the glomerular barrier a little bit or block filtration or block reabsorption, that sort of thing. It's just trying to approach it step by step. But I think absolutely. So I think if we can validate that this is really happening and there is more in the urine, than in the blood in a larger sample, I mean, these are the directions to actually make this better.
Is it correct that most circulating DNA will be negatively charged? Is that correct?
So I don't think that we absolutely know that, because the reason I say that is the current model is that it's wrapped in histones.
Yeah.
And so what the overall charge of that particle is, I don't know, and I'm not sure anybody knows. But I think it would be correct to say that the DNA that's going across the glomerular barrier is very likely not in histones, and it's free, and it should be negatively charged. I agree with that. Yeah. Scott?
I guess I got two questions. So the first question is, what kind of sequencing technology are you using to analyze these 30 base pair fragments of DNA?
Yeah, so because they're so small, it's a long story, but the long story short is we went to the ancient DNA field where they get DNA out of fossils and like woolly mammoth, that kind of stuff, and they had come up with certain protocols that are basically single-stranded DNA ligation based, and that's what we ended up using. Or we sort of modified that slightly and applied it to here, and that's what you need.
And then the second question involves the machine learning techniques that you're using. So you're applying that to these temperature measurements over time to try to pick patterns there. But can you envision a scenario where you're measuring multiple analytes or getting multiple biomarkers you're looking at simultaneously in real time? Would that increase the power of those sort of machine learning algorithms?
Yeah, that's basically the idea. And so right now, so far, the machine learning that we actually applied in our research has actually been to multiple it's multiple data threads, essentially, from the electronic health record. Like, can you take medication data, lab data, those kind of data sets? So absolutely. I mean, I think that's the idea, is that if you build a model with orthogonal types of measurements, that ultimately that'll be more predictive. And then that's where we're certainly headed. But again, I want to just emphasize that hopefully from that, we reduce this to a smaller number of measurements. I expect that that's what will happen, and also make it more practical.
More questions? Thank you very much.
Sure, welcome.